Hi everybody, this is Dr. Udwak Neuro Academy. Today we'll be talking about the physiology of micturition. Micturition is a process by which urine is voided from the urinary bladder. It is usually a reflex process. However, in grown-up children and adults, it can be controlled voluntarily to some extent. To understand the concept of micturition, we must understand the organs and the structures that are involved in the micturition process. The kidneys are the site where urine is produced. The urine actually travels down these muscular tubes called the ureter into the bladder. The bladder stores the urine. When needed, the bladder will contract and release the urine. The urine will travel down the urethra and then become voided. The bladder actually consists of the body, the neck, and the internal urethral sphincter. The smooth muscle forming the body of the bladder is called the detrusor muscles. It is formed by three ill-defined layers of smooth muscles, which are the inner longitudinal muscle layer, the middle circular layer, and the outer longitudinal muscle layer. Now, there are two important sphincters which plays a significant role in holding urine in the bladder. These are the internal urethral sphincter and the external urethral sphincter. In males, between the sphincters is a prostate gland which plays an important role in sperm production. Now, when there is a prostate hyperplasia, it could actually result in the obstruction of urine flow. Now, let's talk about the receptors that are found within the urinary tract that play an important role in micturition and also that plays a significant role in holding urine. The receptors found on the bladder include the muscarinic receptor and the beta adrenergic receptor. We can also find the alpha adrenergic receptor on the internal urethral sphincter. And on the external urethral sphincter, we can find the nicotinic receptor. Now that we know the structures and the receptors involved in micturition, let's look at what controls these receptors and structures. What actually controls the structures and the receptors involved in micturition is actually the nervous system. Now, the central nervous system includes the brain, the brainstem, and the spinal cord. The brainstem is made up of the mesencephalon or the midbrain, the pons, and the medulla. The most important structure in the brainstem that is involved in micturition is the pons. The pons is actually where the micturition center is located. Then we have the spinal cord, which has its various segments, like the cervical segment, the thoracic, the lumbar, and the sacral segments. Now, signals from the spinal cord will be sent to the urinary tract via the nerves. Now, let's look at the three important efferent nerve fibers. The first nerve comes from the sacral segment of the spinal cord. This nerve is actually known as the pelvic nerve, which is a parasympathetic nerve. It is actually not under our control. The pelvic nerve releases acetylcholine. When acetylcholine binds to the muscarinic receptor on the detrusor muscles, it will cause contraction of the detrusor muscles. The second nerve coming out of the sacral segment is the pudendal nerve, which is a somatic nerve. This nerve is actually under our control. The pudendal nerve also releases acetylcholine and acts on the nicotinic receptors found on the external sphincter, causing the contraction of the external urethral sphincter. Now, the third nerve is actually part of the sympathetic nervous system. The preganglionic fibers of this nerve arises from the thoracic segment and synapses at a ganglion called the hypogastric ganglion. It sends neurotransmitters that pass on the impulses to the postganglionic fibers known as the hypogastric nerve. The hypogastric nerve releases the neurotransmitter called noradrenaline. When noradrenaline binds to the beta 3 receptors on the detrusor muscles, it causes the muscles to relax. When noradrenaline binds with the alpha 1 receptors, it causes the contraction of the internal urethral sphincter. So we can say that the sympathetic nerve is responsible for urine retention, whereas the parasympathetic nerve is responsible for voiding or peeing. Also, there is a fourth neuron, which is an afferent nerve. It is a sensory nerve fiber that comes from the detrusor muscle and is stimulated when the bladder is actually stretched. When the bladder is empty, the walls are not stretched. So the sensory pelvic neurons will send slow impulses to the sacral region of the spinal cord. Now, this neuron will actually stimulate the hypogastric nerve from the thoracic and the lumbar region. When the hypogastric nerve is stimulated, it will stimulate the alpha 1 receptor in the internal urethral sphincter, causing the internal sphincter to contract. Also, the stimulated hypogastric nerve will act on the beta 3 receptor to cause relaxation of the detrusor muscles. The brain and the pons also know when the bladder is empty, so that they send signals to the thoracic level to stimulate the hypogastric nerve so that we can actually retain our urine. Also, there are some signals sent to the sacral level that actually inhibit the pelvic efferent nerve at that level. There are signals too that will stimulate the pudendal nerve, which is under our control. When the pudendal nerve is stimulated, it will stimulate the nicotinic receptors on the external urethral sphincter, causing contraction. When we have a full bladder, it will cause the stretching of the detrusor muscles. Now, we have sensory nerve in our detrusor muscles called the pelvic nerve. When the detrusor muscles become stretched, it stimulates the pelvic nerve, increasing the firing of the pelvic nerve. The pelvic nerve sends a lot of signals to the sacral region of the spinal cord. The signals bypasses the thoracic segment, goes to the pons and stimulates neurons in the pontine micturition center, which will inhibit the hypogastric sympathetic nerve. When the hypogastric nerve is inhibited, it causes the contraction of the detrusor muscles and relaxation of the internal urethral sphincter. Also, the neurons coming from the pons micturition center will stimulate the pelvic efferent nerve to cause contraction of the detrusor muscles by stimulating the M3 receptors. Also, neurons coming from the pons micturition center will inhibit the pudendal nerve, causing the relaxation of the external urethral sphincter. So generally, there is contraction of the detrusor muscles and relaxation of the internal and external urethral sphincter, which results in voiding. Now, when we begin to void, the voiding reflex begins. During voiding, the sensory afferent fibers will keep sending impulses to the sacral region of the spinal cord. It stimulates an interneuron on that level, which will connect with the parasympathetic efferent nerve fibers. When the efferent fibers are stimulated, it will cause the contraction of the detrusor muscles. So in actuality, it looks like a loop. Thank you very much. I hope you find this lecture interesting.